Hello students, welcome back to the lecture series in photosynthesis. Now in today's lecture, we are going to start with the concept of photosynthetic pigments. इससे पहले कि हम photosynthetic pigments जानें और उनकी classification क्या होती है, we are going to learn mainly about the basic of photosynthesis, which is clear to almost everyone, right from the day when you start preparing for your medical examination. Now, photosynthesis is basically a physiochemical process. Now, the thing is, in this physiochemical process, the light energy, is actually being absorbed by the photosynthetic organism. Now, why I have not written the word plant, you will understand it very soon, because concept of photosynthesis in our discussion will not only be limited to plants, we will also discuss bacterial photosynthesis at a later stage. So photosynthetic organisms, they actually absorb the light energy, and that light energy is first of all being used up for a series of reactions or a type of reaction which we call as the light reactions. Since they are directly dependent upon light, they are called as light reactions. And then the second phase of photosynthesis is going to be the dark reaction, right? Now we have the light reaction and we have the dark reaction. Now, first of all, we can also understand that what is the basic difference or purpose of the light reaction. The light reaction is mainly responsible for producing two kinds of assimilatory power, which are known as ATP and NADPH. So they are called as the assimilatory power. Why assimilatory power? Because they will actually help in preparing the food, which will be utilized by the photosynthetic organism. Now, in the dark reaction, the assimilatory power of the light reaction is being used up, and the CO2 may be absorbed from the atmosphere. And ultimately, the carbon of the CO2, the carbon which are available in CO2, they are converted into the triose phosphates followed by the glucose. In some cases, and especially in case of plants, we may call this particular procedure as the step of carbon fixation. So carbon fixation can also be the another alternative word which can be used for the dark reaction. But yes, just like for preparing food, you need to arrange for the, let's say maybe the LPG cylinder or your different components. So just like preparing food requires certain prerequisites which are to be arranged first, similar to that, photosynthesis, the light reaction is actually the phase where all the preparations are being done. And then in the dark reaction, since it is not dependent upon the light directly, therefore it is also called as the light independent compared to light reaction, which was called as the light dependent reaction. So the dark reaction is also known as carbon fixation, and there mainly the carbon fixation or the food preparation happens with the help of whatever assimilatory power has been prepared in the light reaction. Now, as far as the concept of photosynthetic organisms is concerned, direct questions in CSR Part B may also be asked from this area, that photosynthetic organisms can be classified as oxygenic and anoxygenic depending upon whether O2 is evolved in the photosynthetic procedure or not. So O2 is evolved or O2 is not evolved, yani कि gas other than oxygen is evolved. So no O2 evolved. No O2 is liberated or no O2 evolved. So do not think that the if O2 is not evolved, no other gas will be evolved. Gases other than oxygen are actually evolved in that case. Like sulfur may be evolved in case of the sulfur bacteria, right? So O2 is evolved, then it is called as the oxygenic photosynthesis or the oxygenic photosynthetic organism. And if no O2 is evolved, it is called as the anoxygenic one. Now, the oxygenic organisms or the oxygenic photosynthetic organisms have been divided into, I mean, they have been dispersed across the prokaryotic system as well as eukaryotic system, meaning we have examples of prokaryotic oxygenic organism as well as eukaryotic oxygenic organism. In prokaryotic system, you have to remember the example of cyanobacteria, which can be a direct question. While in case of eukaryotes, we have to remember about the plants and the photosynthetic protist. On the other hand, N-oxygenic, that is no oxygen is evolved, such photosynthetic organisms are mainly grouped into the prokaryotic organisms, and green and purple photosynthetic bacteria are one of the best examples belonging to that. Moving to the next information, we are going to talk about the photosynthetic pigments. Now, the photosynthetic pigment, the first thing that comes to our mind is the word chlorophyll. And everybody knows that in a basic chloroplast cross section, there are certain stacks like structure, single coin like structure. There's a single flattened coin like structure called as thylakoid. And a group of thylakoid is basically called as the granum, right? So the cluster or the stacking of thylakoid results in something called as the granum, right? Which is a group of thylakoid or a stack of thylakoid. Now, when we look into the ultrastructure of the thylakoid membrane, across the thylakoid membrane, we can easily see that chlorophyll molecules are embedded in the proteinaceous matrix, right? So they are being embedded inside the thylakoid membrane. This is sometimes also referred to as the light harvesting chlorophyll complex, or in some basic books, you may also find the word called as the LHC, which stands for the light harvesting complex. So we may even use these two words for the purpose of explanation of the LHC or the light harvesting complex and the light harvesting chlorophyll complex, one and the same thing. But what if you look at the detailed ultrastructure of a chlorophyll molecule? So we will be discussing that. But before discussing the ultrastructure of chlorophyll, let's come back to some of the basic properties from where part B CSR questions are coming directly. Chlorophylls, first of all, they are considered to be the lipid soluble pigment. As clearly seen by their hydrophobic nature, they are able to penetrate themselves or they're able to arrange themselves across the thylakoid membrane. That's only possible when they are lipid soluble because light dissolves light as per the rule of chemistry. The next property that we have, they are light absorbing. Obviously, it's light absorbing pigment, but along with that, it's a green pigment. It's a green colored pigment, right? The next thing says that it contains polycyclic. Poly means multiple, right? And cyclic means cyclic ring-like structure. So you can see that there are multiple cyclic rings which are available in the structure. So it is polycyclic planar that it lies in the plane of the paper and tetrapyrrole. So it's a polycyclic planar tetrapyrrole ring structure. What is the meaning of tetrapyrrole? Tetrapyrrole means four pyrrole rings, right? So the four pyrrole ring. So there are four pyrrole ring which are easily visible to us. That is the ring number 1, ring number 2, ring number 3, and we have the ring number 4. So there are four pyrrole rings, and that's why it's called as a tetrapyrrole. And remember, it's a cyclic tetrapyrrole, not a linear one, right? As you can easily see that these four pyrrole rings are arranged in a cyclic manner to form a cyclic structure. Coming on to the component, we can easily see that there are two main parts in this particular thing. What are the two main parts that we have over here? One part which is known as the porphyrin head and the other part which is called as the hydrocarbon tail, right? So we have the porphyrin head, we have the porphyrin head, and we do have the hydrocarbon tail. Now the thing is, let's have a look at this porphyrin head and the hydrocarbon tail properties. So porphyrin head has been accounted for the main light absorption. That means if somebody asks us that which part, there are two major parts, the porphyrin head and hydrocarbon tail, which part is actually responsible for the absorption property of chlorophyll molecule, then the light absorption is a property of the porphyrin head. And everybody knows that with the help of the structure easily visible to us, we can say that the porphyrin head is basically a tetrapyrrole, that it contains four pyrrole rings which are actually joined with each other with the help of the Mg2 plus ion, metal ion at the center. On the other hand, let's talk about the hydrocarbon tail. The hydrocarbon tail is 20 carbon in length, and the most important role for this hydrocarbon tail is that it helps or it secures the chlorophyll molecule in the thylakoid membrane. These questions have been asked in the previous year examination and forms an important part of our discussion. So it secures the chlorophyll molecule in the thylakoid membrane. That is the main role attributed to the hydrocarbon tail or the phytol tail, which is also an alternative name. So let me write it over here. This hydrocarbon tail in some books may also be written as the phytol tail, right? So we have the porphyrin head and we have the phytol tail. Porphyrin head is mainly responsible for the light absorption, and the hydrocarbon tail or phytol tail helps to secure the position of the chlorophyll molecule in the thylakoid membrane. Now, don't worry about the structural aspects, whatever is written over here, that pyrrole ring 2, that this pyrrole ring 2, if it contains a methyl group, then it becomes chlorophyll A. And if it contains chlorophyll, this aldehyde group, CHO group, then it becomes chlorophyll B. We'll learn about that definitely. So one thing is for sure, if somebody is asking you a question in the examination that out of the two parts, which is actually a cyclic tetrapyrrole, so it's visible to us that the porphyrin head is a cyclic tetrapyrrole. On the other hand, the hydrocarbon tail is just a linear molecule, which is helping us in securing the position of chlorophyll molecule within the thylakoid membrane. So this is just not the end of our discussion about chlorophyll. We'll have to look at the structural aspect of the chlorophyll molecule, and let's go into that particular detail. Now, based on this figure, which we can see over here, it's clearly visible to us that there are 4 pyrrole rings, ring number 1, 2, 3, and 4. And mind it that you cannot change the position or representation. Its naming has been done in this particular serial order only, that we are moving in a particular anti-clockwise chain over here. So as based on the figure, you can easily see that the central metal ion visible to us over here is Mg2 plus. There's absolutely no doubt about it. And the nitrogen atom or the nitrogen position from the ring number 1, 2, 3, and 4 is actually holding the Mg2 plus in the center. As discussed earlier also, the porphyrin head is actually responsible for light absorption. Therefore, this point indicates that light is being absorbed by the complex ring of a chlorophyll molecule. On the other hand, the hydrocarbon tail secure the chlorophyll molecule to hydrophobic proteins inside the thylakoid membrane. Absolutely no doubt about it. Now, apart from these 4 pyrrole rings, I hope everybody is able to see this particular ring, which is resembling the 5 carbon ring, and then we use the word called as the cyclopentane, and therefore we have the word called as cyclopentanone ring. And why the number 5th has been given? Because there are already 4 cyclic rings called as the pyrrole ring 1, 2, 3, and 4, and this 5th one, which is resembling the cyclopentanone ring or cyclopentane ring. So this cyclopentanone ring is called as the ring number 5. But a question definitely asked in the examination that the cyclopentanone ring or ring number 5 is fused to which of the pyrrole ring? The question is simply answered based upon this figure. So this becomes clear to us now that the cyclopentanone ring 5th is actually fused to the pyrrole ring number 3. Now, whether the chlorophyll molecule is a chlorophyll A or it's a chlorophyll B depends upon what kind of substituent has been joined to the ring number 2. So if it's CS3 group, then it is a chlorophyll A molecule. And if the same position is being filled by a aldehyde group instead of a methyl group, then it becomes an chlorophyll B molecule structure. So if pyrrole ring 2 contains a methyl group, then it is chlorophyll A. And if it is an aldehyde group, then it is definitely a chlorophyll B molecule. Absolutely no doubt about that should be there, right? Moving ahead with our continuing discussion. Let's try to understand, since chlorophyll is the main pigment, therefore we are giving so much time and dedication to the chlorophyll molecule. So there are 2 main molecules between which we have to understand a comparative account, the chlorophyll A and the chlorophyll B. As indicated by the formula, you can see that there are certain common parts, right? So it's quite easy to remember the chlorophyll A and B structure or the formula. So we have common thing called as the C55 over here and N4Mg. I'm highlighting the common parts for you. So C55 N4Mg, you don't have to, differentiate or learn it as a part of a differential molecule. It's common between both of them. So C55 N4Mg is always going to be there. And the difference which is coming is between this particular part for you, which is the H72O5 in case of chlorophyll A and H70O6 in case of the chlorophyll B. So chlorophyll A, when it is being present in a purified state, so blue-green pigment in its pure state. So this is a blue-green pigment in its pure state, while this is an olive-green pigment in its pure state, which is very important thing. I mean, we are talking about their purified states or even the crystallite state that we are considering. So it's a blue-green pigment. On the other hand, this is an olive-green pigment in its pure state. As everybody knows that chlorophyll A is an essential photosynthetic pigment, while chlorophyll B is an accessory photosynthetic pigment. As discussed in the previous this structural figure that if the ring number 2 contains CH3, then it is chlorophyll A, and if it is a CHO, then it is chlorophyll B, right? So pyrrole ring number 2 contains CH3 group as for chlorophyll A, while pyrrole ring number 2 contains an aldehyde group or CHO group. Now, this last point can also be considered to be a very important information for the examination, that chlorophyll A absorbs light more strongly or more strong absorption in red wavelength than violet blue wavelength, while chlorophyll B shows the reverse of that. So it shows a stronger absorption in the violet blue wavelength compared to the red one, that we have to remember. So this is a comparative account for chlorophyll A and B. And structures, you will have to just retain it in your mind. Nobody is going to ask you to make structures, but formula you should remember, that can be one of the differentiating points, or a sort of multiple choice statements or multiple answer questions in your part C questions, right? Moving ahead to the next thing that we have, chlorophyll types, quite an important thing. Our discussion not only ends over here, because this particular lecture is mainly determined to finish off the photosynthetic pigment part 1 area, where we are destined to cover mainly the chlorophyll aspect or the chlorophyll thing, right? So that's why we are giving so much importance to chlorophyll. So chlorophyll types, if we talk about, depends upon what kind of organism you are talking about. And then it can be either present in the oxygenic photosynthetic organism, or it can be a part of anoxygenic photosynthetic organism. So we have oxygenic photosynthetic organisms and we have anoxygenic photosynthetic organisms. So in oxygenic, we have chlorophyll A, chlorophyll B can also be present, chlorophyll C has been reported, chlorophyll D has also been reported. And how do these molecules mainly differ in their structure? We are mainly concerned because our syllabus mainly includes the difference between chlorophyll A and chlorophyll B. But if you want to understand, I mean, chlorophyll C and D, how these naming have been done. These chlorophyll molecule once again differ in terms of subsequents present on tetrapyrrole ring. So just like for chlorophyll A and B, we remember that the difference was on ring number 2, pyrrole ring number 2. If methyl group is there, it's chlorophyll A, and if the CHO group was there, it's an aldehyde group, the CHO group, then it's a chlorophyll B thing, right? Similarly, C and D have been based upon the different substituents or the groups that are present on the pyrrole rings. The structure of C and D has not been completely elucidated, and we are also not interested in it since it is not a part of our syllabus. But apart from mentioning A, B, C, D, I would like to mention the name of chlorophyll E also, which is not given in most of the books. It's an exceptional pigment, and it has been reported in case of some golden algae, right? So sometimes in part C question, you may also find that chlorophyll E is a pigment found in some thallophytes. So it's correct. Algae actually belongs to a particular classification, which is called as the group known as the thallophytes or the thallophyta, right? So coming to the next thing that we have over here is the anoxygenic photosynthetic organism. What about them? The anoxygenic photosynthetic organisms do not contain chlorophyll. Instead, they contain something which is called as the BCHL or the bacteriochlorophyll. Now, the points which may be asked in the examination pertaining to the BCHL is that the BCHL or the bacteriochlorophyll, it absorbs light at a longer wavelength compared to the normal chlorophylls, right? And the different types of molecules for BCHL may range from bacteriochlorophyll A, B, C, D, and E. Do not worry about bacteriochlorophyll structure, because bacteriochlorophyll structure, I mean, these particular aspects, they are later going to be discussed as a part of something called as prokaryotic photosynthesis. So don't worry about it. Prokaryotic photosynthesis would be a separate a separate lecture of ours, where we will be discussing about the bacteriochlorophyll. That's why we are not interested in understanding the BCHL structure over here. Will understand their photosystem, their component at a later stage. Coming on to the next important part that we have is the accessory pigments, right? So what do you understand by accessory pigment? Accessory pigment, I mean, apart from the main photosynthetic pigment, which is the chlorophyll, and that too among chlorophyll, chlorophyll A is most important, which we will discuss later on while we'll be discussing the photosynthesis mechanism. Accessory pigments are those pigments which are not directly performing the light absorption, but rather they are providing protection to the reaction center chlorophyll, which we will be discussing later on. But the thing is, as far as their absorption is concerned, they absorb the light wavelength where chlorophyll do not absorb strongly. That's quite important point, right? So their absorption of the light is actually where the chlorophyll do not absorb strongly. Whatever range chlorophyll is absorbing, whatever in the range like 400 to 700 nanometer for the chlorophylls, their that is their strong absorption hold. But the ranges outside that, the accessory pigments might be concerned with that particular absorption. We will be understanding the wavelengths also when we will be progressing our discussion today. So there are two types of pigments called as the carotenoids and the phycobilins. So accessory pigment not directly involved in the light absorption. They are actually they are protecting the main reaction center chlorophyll from damages. Do not worry, I will write every single point when we will be discussing carotenoids and phycobilins in detail in the next lecture. For today, what we are interested in is that carotenoids actually maintain a particular thing which is called as the lipid soluble pigment nature. On the other hand, we have the phycobilins, which is a water soluble pigment, right? So accessory pigments, two broad categories: carotenoids versus phycobilins. Carotenoids actually form the lipid soluble pigment, whereas the phycobilins form something called as the water soluble pigment. Now, carotenoids can be further distinguished into xanthophylls and carotenes. We will understand what can be the basis of that thing in the upcoming slide, where you'll see that this particular classification of xanthophylls and carotenes is mainly based upon their structure. So, this classification of carotenoids into carotenes and xanthophylls is mainly based on their structure. Now, as far as these accessory pigments are concerned, we will initiate our discussion mainly about the carotenoids first, and in the upcoming lecture, we will be taking more details about carotenoids followed by the phycobilins. Now, carotenoids, if we talk about, even before classifying them into the carotenes and the xanthophylls, as we have written in this particular thing. So carotenoids is our first point of discussion, and there are two types of carotenoids like the xanthophylls and the carotenes. But what can be the basis of such a classification? For that, I have represented certain structures in front of you. So carotenoids, some general points from which questions can be asked directly in the examination. They are long chain, definitely, whether it's carotene or whether it's xanthophyll, they are long chain molecules, as you can easily see over here. They are conjugated hydrocarbon. Yes, that is also true. And I hope everybody understand that this is something sort of a conjugation that we are representing. And conjugation means the presence of alternate single and double bonds, right? So alternate single and the carbon-carbon double bond, which is actually seen here. That's what we mean by the conjugated hydrocarbon. Usually the number of carbon in them is 40 carbon, and therefore they are also called as C40 terpenoids, right? So they're often called as what? C40 terpenoids is their chemical nature, and they are formed by condensation of or joining of 8 isoprene unit. For your knowledge, I must tell you that one isoprene unit or one isoprene technically has 5 carbon atoms. Therefore, in order to form the 40 carbon atoms represented over here or in the molecules of the carotenoid, at least 8 isoprene units have to join together, since one isoprene is equivalent to a 5 carbon system. Now, the thing is, absorption spectrum may also be asked in the examination. It ranges from 400 to 500 nanometer compared to normal chlorophyll, where the absorption spectrum may lie somewhere in the between the entire pair region, and they are more concentrated toward the red and the violet blue. We will discuss it in the upcoming lecture, where when we will be discussing about the absorption spectrum. Don't worry about that thing, right? So carotenoids can be classified into carotenes or xanthophyll based on structure. As you can see that carotenes are purely hydrocarbon, absolutely no oxygen is there, whether it's lycopene or beta carotene. You will have to remember these names. So lycopene and beta carotene are actually carotenes, and they're simply hydrocarbons without any oxygen. So lycopene, beta carotene belong to the carotene subcategory of the carotenoid molecule. While the xanthophyll thing, the list is long, but it's quite easy to remember also. But The structural difference that you can easily see is the presence of oxygen. I hope everybody can see the presence of oxygen is there, and this clearly demarcates the carotenes from the xanthophylls. So xanthophylls contain oxygen. On the other hand, carotenes are purely hydrocarbon, absolutely no oxygen over there, right? So we have certain examples over here called as the viola xanthan, then we have the zeaxanthan, astaxanthin, spirulozanthan, neoxanthin. Now the key word to remember, I mean, if towards the last you are having these words like the xanthan word, as you can easily see, so xanthan clearly indicates. So as soon as you see the word xanthan, quickly understand that it's a xanthophyll. Absolutely no doubt about it. But the names which may confuse you are terms like lutein, right? Fucoxanthin once again and canthaxanthin. Word xanthan is coming again and again, and it clearly indicates that we are talking about the xanthophyll, right? Lutein, please remember about it. Lutein is quite an important thing which might confuse you. So lutein is a xanthophyll, just like the others which have been named, while lycopene and beta carotene are clearly belonging to the word carotenes, right? Beta carotene will not confuse you because it's simply a carotene. Remember, the word lycopene is a carotene and lutein over here. So there are two words starting with the letter L, lycopene coming in carotenes category, and the lutein, on the other hand, coming in the xanthophyll category, right? So this is initiating information about our accessory pigment, and that will be all for today.